Hi, today I wanted to talk to you about the experiment of study in batch reactor kinetics. A batch reactor looks like this, and it typically has a stir, inlets for the reactants, and a jacket around the outside so that you can control the temperature, as well as a vent to the system so that you can control the pressure. Batch reaction in industry is used for multiple products so that you can use the same equipment to produce multiple products. Small scale production, which means you can have a batch reactor ranging from 5 gallons to 8,000 gallons in volume. High conversion, which is time variable, so if you're looking for a specific amount of conversion, you can usually leave it in the batch reactor longer in order to achieve that conversion. It does have high labor costs typically associated with it, which is a downside. But another upside is that you can control the temperature using that cooling jacket on the outside and the pressure pretty well using a ventilation system. The purpose of this particular experiment is to simulate the batch reaction, determine the rate law governing the reaction, determine the rate constant and activation energy, illustrate the temperature dependence of reactions themselves. Important materials in this experiment are sodium hydroxide, crystal violet, DI water, colorimeter, stirring rod, cuvettes, and the 50 milliliter beaker. When doing this experiment, you want to perform it with proper safety, and you would like to have goggles and gloves on and avoid spilling the NOH because it is caustic, so it can cause skin irritation, and avoid spilling crystal violet because it can cause a biological stain, which isn't fun to have on your skin or your clothes. As you can see, the guy here is dressed properly in the side of the screen with the check mark and improperly on the side of the screen with the X. Rate law determination is done first by calibrating the colorimeter. There is a file already on the computer for this experiment, so make sure you find the correct one. Prepare 4 cuvettes with the known crystal violet concentration, which is done so that you can later on use it to correlate the colorimeter readings with the concentration of your solution. And this is going to be done using DI water and crystal violet, not the reactants. And then you're going to prepare the 10 milliliter components for the reaction itself, specified by the lab guide, and you're going to pour them into the 50 milliliter beakers simultaneously. Here is the video of us doing that. You can see here we have the magnetic stirrer on the bottom that's simulating stirring the reaction, and our little 50 milliliter beaker is actually simulating our batch reactor itself. Furthermore, in the rate law determination portion of this experiment, you're going to put the solution into the colorimeter every 2 minutes. I recommend getting it ready to put into the colorimeter a little early so that you can get the reading on the exact 2-minute mark, but you want to also make sure you take it out of the colorimeter to prevent it from heating up too much because that will mess with your kinetics of your reaction. Then you want to collect data points at each 2-minute mark and then repeat it using a different NaOH concentration for the later part of the experiment and for the rate law determination step with respect to NaOH. For the rate constant and activation energy determination portion of the experiment, you're going to select the correct experiment from Vernier. So again, there's a file already created for you. Set up the temperature probe and colorimeter, and prepare 4 samples as specified in the lab guide. I do recommend that someone at the beginning of the experiment, while everybody else is working on the first portion of it, prepares these samples because they do need a time to chill to a certain temperature in the refrigerator. Then you're going to conduct the first trial and be ready to time the reaction, and then repeat it for the remaining trials and at different temperatures, and then export your data to Excel. And those are my recommendations for the experiment as performed, and you are done with the experiment.